Hello, thanks for joining us today. I'm Carrie Burton, your host for today's webinar. And with me today is Tim Batten and Tim Smith to discuss combining Raman spectroscopy for richer science. Before we begin, please note that you are on mute and will remain in listen-only mode throughout the webinar. However, we encourage you to submit your questions or comments at any time during the session using the control panel on the right side. We will be having a Q&A at the end and hope to answer as many questions as possible. And we are recording today's webinar and we'll be sending the replay to you via email. And now I'd like to welcome Tim Batten, Senior Application Scientist at Renishaw plc in the Spectroscopy Products Division. Hello, Tim.
Thanks very much for the introduction, Carrie, and thanks everyone for attending today. I'd like to talk to you about combining Raman spectroscopy for richer science. So Raman spectroscopy itself is a really powerful tool. You can do lots of things with it. You can measure lots of different sample types without any sort of preparation, and it gives you information on the chemical, mechanical structure, and crystal properties of materials, all really useful things to solve general scientific problems. But in the normal circumstances, Raman alone is rarely enough to give you a really firm conclusion to solve a complex scientific problem. So we often provide Raman systems which are used in combination with other techniques, so for instance with SEM or AFM. And recently we've developed a tool called Correlate, which enables you to take other measurements from any wide range of techniques, import them into our Raman analysis data so you can see the data side by side and do combined analysis. And to be honest, this is where Raman really stands up. It provides you lots of useful information, which is really complementary with other techniques and lets you form very firm conclusions about your work, ultimately leading to good science. So here I just have a summary slide, a summary of my talk. I'm going to start off by talking about the latest developments in combined techniques. So this is looking at sample topography with our LiveTrack technology, or combining Raman with AFM or SEM or nanoindentation. I'm going to briefly give an overview of each of these techniques, and then I want to give a few basic examples just to give you sort of a refresh of what can be done and to make you think about what can be done in your research with the techniques. I'm then going to go on to do some advanced case studies, and here today I'm just going to talk about two different ones. So the first is looking at mineralogical sections and basically correlating SEM images and Raman measurements of these sections to try and get better understanding of the samples and the systems in place. The second case study is looking at rhenium disulfide flakes. Rhenium disulfide is a novel 2D material. And here we're just going to look at characterizing different properties of the rhenium disulfide. So that might be layer thickness or other properties, using a wide variety of techniques to show that we can do this and how we would go about doing it using our Rayleigh inVia Raman microscope. This is just a section slide, but what I really wanted to highlight here is some of the many possible combined systems you can get with your Rayleigh Raman system. So on the left-hand side, we have a Rayleigh SEM-SCA Raman system, and that connects to an SEM, and that allows you to do in situ measurements inside the SEM chamber. In the middle, we have an inVia connected to a Hydrotron and now Brooker nanoindentation system, and again, you can do the simultaneous measurements, so you can do nanoindentation and Raman at the same time, so you can measure the various different properties of material as you're directly indenting it. And on the right hand side we have an inVia coupled to an inverted microscope, and on that inverted microscope is an AFM, and this lets you do a number of different co-localized measurements. So first of all, you can do the Raman, you can do the AFM, and then on the inverted microscope we have epifluorescence, so you can do them all together on the same sample without having to move it. So first I'd like to talk about combined topography measurements using our LiveTrack technology. LiveTrack is something that is a patented Rayleigh technique, and it's an autofocus and sample tracking technique. So this enables you to do Raman imaging of non-flat rough samples. And as you can see on my animation on the right hand side, effectively what we do during the measurement is we use our Z stage to adjust the sample to maintain the focus of the surface during the measurement, and this is achieved using optical feedback based on the laser beam. It's very fast. It means your Raman measurements are always in focus. You can also use it in other modes, so we can use it in an optical browsing mode. So when you're looking at a sample under the microscope, your sample will remain in focus. You don't have to keep adjusting the Z to compensate if your sample's sloped or something like that. Because we're moving the stage up and down, and we record the position of this stage, this allows us to get topographic information from the sample. So we're able to see what the change in height is of the roughness, things like that. But fundamentally, this is a really, really nice tool for measuring samples with complex geometries. So you can measure anything from a slope slide down to macroscopic objects. So one of the examples I'm going to look at in a minute is a rat mandible. You can also measure things like battery electrodes, which are very, very rough. And again, I'll show you that in a second. So for my first LiveTrack example, I wanted to show this one. This is a rat mandible. And the reason I really like this example is because it shows something which would just be impossible to do without this LiveTrack technology. So here we've got a truly macroscopic object. It's approximately 19 millimeters by 70 millimeters. Across the object, we see significant changes in height, so a total of 1.85 millimeters. Okay, so if you tried to do this on a Raman system without LiveTrack, you would basically be forced to section the mandible and measure it. Whereas with LiveTrack, we can just put it underneath, no sample preparation, and go. And you can see this in the structure itself. So we've got lots of substructure in the mandible. So the topography image shows how it changes. So I can, for instance, circle this is the tooth and root region here. And we can see we've got a nice tracking over the curved geometry of that piece. Obviously, at the same time, we're collecting Raman data. And here, I've just created a simple Raman image, and this just shows the mineral to matrix content of the bone. So when I'm talking about the matrix, I'm talking about the organic components. And when I'm talking about a mineral, I'm talking about the apatite, so the presence of the phosphate band. And this gives us an indication on the variation in the structure. Clearly, there's significant variations across the whole mandible, and this is due to the different functional parts of the structure itself. But yeah, this is a really fantastic image, and I really love it. My next example is a battery electrode. This shows another circumstance where LiveTrack is really powerful. So in this case, we've got a surface which is very rough. So if you conduct a Raman map over this surface, you'll have areas where you'll go in and out of optical focus, and that will start to present artifacts in your results. And in most cases, the way you'd do this, get around this, is to compromise, go to a lower magnification, lower NA lens, and then that affects your run performance. But in the case of LiveTrack, we don't need to worry about it. So what happens is, again, we move the sample up and down to correct for this change in focus in real time. What you can see from the optical image here, and I'll just circle a region, is a region which is out of focus. And you can see this region on the corresponding LiveTrack image here. So we can see the two dips, again, giving us confidence that this is all working fine as expected. Looking at this sample, we can see that the change in height is around 30 microns. So again, this is bigger than the depth of field of the sort of objective lenses you'd like to use for these experiments. And on the next slide, I'm going to go into a bit more about the science of this sample. So just a brief overview on these sort of battery electrodes. So in this case, you get electrical chemical reaction in these devices, and it occurs in the porous electrodes, which primarily consist of transition metal oxide mixed with conductive carbon and a binder. And the binder and the carbon form porous networks, so you can get the active material moving in and out of that network. In this particular electrode, we can measure with Raman, and we see two primary forms of carbon. So we see natural graphite in red and carbon black in yellow. And these have very distinctive spectra. And we can plot this over a map of the same area as in the previous slide. And you can see we've got a mixture of carbon black, natural graphite, pretty much spread evenly about the surface. Carbon black accounts for 75% of the material, and natural graphite is about 25% of the material. Going to the next slide, we can start to combine all this information together. So the a topography image is shown here, and in this case, we basically just have the change in height. And on that topography image, I have shown the carbon black and the natural graphite. So in red, we have the natural graphite, and you can see that that tends to occur on the peaks of the topography, whereas the carbon black and yellow tends to occur in the valleys, and this is of course related to particle size. So the carbon black particles are much smaller than the graphites, they don't tend to stick out, and they tend to infill the areas. And obviously this is a useful piece of information which you wouldn't be able to get using Raman on its own. My final LiveTrack example is taking this technique and pushing it a step further. So here we're looking at dynamic samples. So you can imagine having this autofocus, it doesn't just mean we need to look at static things. And it's really nice that it keeps the sample in focus all the time. So we can measure dynamic systems, i.e. ones which are changing due to reactions. So that could be because they're being heated or cooled. There are chemical reactions going on there. You've got fluid that's evaporating, all sorts of things. Here I've run a very simple experiment. So in this case, I've taken a polyethylene pellet, I've placed it in a crucible, and I've heated it to 200 degrees, then cooled it back to room temperature. And during that measurement, I've used LiveTrack to maintain the focus on the sample and collected Raman data at the same time. So here we can just see a simple change in position graph against temperature. So my pellet starts at this particular height. As I heat it, it expands till we get to about 150 degrees where it starts to melt. When it melts, the pellet changes volume to occupy the whole crucible. So we see a massive change in the height, in this case a height change of 1.5 millimeters very quickly. Then as I cool the pellet, you can see again it starts to shrink and contract until eventually we reach a pretty much standard consistent height when it's reformed and recrystallized here at about 100 degrees. At the same time, I collected the Raman data, so we can see here this is the initial data point. So we've got the orthorhombic band, which gives an indicator of the phase. So as we heat the sample up, we can see we've generally got the same spectrum. It's getting a bit hotter, so the peaks are broadening out and we're starting to lose some of the crystallinity. As we go above 150 degrees, so here I've got a point around 170, you can see that this orthorhombic band's starting to decay. By the point we get a bit further, you can see it's gone in its entirety, so we now just have liquid. This continues, we let the sample cool down. As it cools and recrystallize, we get the band back again, very similar to the above. And then we let it go back to room temperature and we have a very similar spectra to the above spectra. So what I really want to show you here is we're able to track the changes of the material even though it's moving massively in this temperature cell. And this is something which you really need this sort of fat, quick autofocus technique to do. Now I want to move on to Raman AFM. The nice thing about AFM is you can characterize and image things which are very different to Raman spectroscopy. So things like topography, adhesion, elasticity, potential, all with very, very high spatial resolution. So relating these properties to the stuff you get from Raman is very useful. In addition, using an AFM and a Raman system in combination lets you do something called tip-enhanced Raman spectroscopy, and that can be used to provide Raman information with really high spatial resolution, so on the order of 20 nanometers. We work with a wide range of AFM vendors, and they can all be coupled to our Raman system, and they all have their various different pros and cons depending on the sort of samples you want to look at. So I'm going to run through a couple of AFM examples now. So here is an example which has been done co-localized. So we've used an AFM to get the topographic information from a glioma cell, and that's the image on the left-hand side. So you can see the cell body in blue, and then we've got the nucleus and other unidentified subcellular structures are shown in pink. Okay, if we then put the same cell on the Raman system, use our co-localization software, we're able to map the same area, and here we've done a volume image of the cell, and we've been able to isolate the nucleus simply by looking at the nucleic acid band around 800, and we've also been able to isolate other substructures. So in this case we see lipid droplets and membrane-bound organelles. And what I really want to show you here is by using the two techniques in conjunction, we get more information about the sample. My next example is carrying out tip-enhanced Raman spectroscopy on a semiconductor wafer. So this is a silicon wafer which has been doped with germanium, and the wafer was ion implanted from the top. So what happens is the top say 100 nanometers of the wafer has differing levels of germanium in it, and we want to do TERS to try and identify what that germanium concentration is. So here we're running on a cross section of the wafer, and we were able to plot the germanium peak position and germanium intensity against line distance. So here we can see a gradual decrease in the germanium intensity as we go towards the bulk of the wafer, and we can also see how the germanium changes, peak position changes, which is an indicator of the relative strain. in the germanium structures in this wafer. The same can be done with the silicon, so we can again see how the silicon changes as we have more germanium-rich regions and how that impacts its mechanical properties. Now I'd like to briefly talk about Raman SEM options. So SEM's used everywhere for a wide range of applications, primarily for imaging. A significant number of SEMs also have EDS systems mounted on them, and this enables you to get elemental information from a sample. But Raman really offers some excellent complementary information here, because you get your lovely image, high magnification image with your SEM, you get the elemental information with EDS, but sometimes it doesn't solve the problem. So Raman provides the chemical information that enables you to sort of link all these things together and get you a better understanding of your sample. So I'll show an example of that in a moment, and we'll talk about it a bit further. But here I just really want to highlight that you can collect Raman spectra inside the SEM using our SEM-SCA technique. You can also do mapping and imaging, and this instrumentation can be coupled to a range of SEMs from all the major manufacturers, so Hitachi, JL, T-Scan, and Zeiss. So here I have a simple example of why it's useful to have Raman in conjunction with SEM and EDS. So on the right hand side here we've got an image of a series of fibers, and on top of those fibers are a number of different particles, all of different chemical composition. Now EDS enables us to get elemental composition of these particles. So for instance, it will be very good at identifying that we have silicon carbide here. However, if we go to the PTFE particle, when we do EDS, we will just see elemental carbon. Now, that's clearly not that useful. However, if we take a Raman spectra from the same particle, we will get a Raman spectra of PTFE, which we can just simply run through a database, match it with PTFE, and we've got a confirmed chemical identification. The same to some point is true with the chalk. So in this case, it's calcite, calcium carbonate, and we would be able to get the elemental information from EDS, but we wouldn't be able to identify which polytype of calcium carbonate is, so we wouldn't know if it's, for instance, calcite or aragonite. However, Raman is able to give us that crystallographic information, so we're able to get a positive identification. And here I just wanted to show you an example of Raman mapping in the SEM. So here we've got an indented silicon wafer, and we've got the SEM image, and this is the corresponding Raman image. So here we're looking at the silicon peak position, which is an indicator of strain and stress. So basically we're able to see correlations between the various different shapes in the Raman image to the SEM image, and we're able to just get a little bit more information from our sample. The last combined technique I'd like to talk about is combining Raman and triboindentation. So here we're using Raman, again, using Raman spectroscopy to give information on the composition of materials and their stress and strain, etc. But it doesn't give you the mechanical properties and the tribological information we can get from nanoindentation. But combining these two techniques together allows us to directly correlate those properties. So we can correlate the mechanical properties with chemical analysis and do it in situ. So for this example, I've got 3 DLC films. They're all deposited on silicon, and they were nano-indented. The central figure shows the Raman spectra for each of these films. They have D and G bands, standard for DLC type films. You can see that A in red and B in blue are quite similar. Film C in black is a little bit different. So there's 2 key differences. One, you can see the presence of the silicon second order band, and that's because the film itself is very thin, only 40 nanometers. The other thing to note is it doesn't have a very strong D band, and that will affect its mechanical properties. If we now look at the load indentation depth figure on the right hand side, you can see how these 3 different films vary. So the film A and film B have quite similar properties. You can see that to indent film A, we need to apply more force than film B, so it's clearly a harder film. With regards to film C, though, we can see quite a bit of difference. So it's a lot softer, so we're getting much deeper indents with the same level of force. But also on the unloading curve, we see an elbow here. So it's not quite elastic in its recovery. So something is going on here. And what we suspect is happening is you're getting a transformation in the silicon phase underneath the film. And basically, because this film is so thin, it's not really protecting the silicon underneath. So we're measuring just as much of the silicon changes as we are of the film changes. Now I'd like to talk about my first case study, which is doing correlated SEM and Raman measurements of a mineralogical section. First, I'd like to introduce Correlate. So this is some new software functionality we've got in Wire 5.3 onwards, and it enables you to perform measurements on inVia at the same location as measurements we've performed on another microscopy type system. And to do this, we have a coordinate manager. This allows you to take two relative coordinate systems and basically transform them to form the same system. So we can take a SEM and with its own coordinate system, and we can take inVia with its separate stage coordinate system, convert them to one system so we can compare the results and do measurements in the same place. This is achieved using the batch mapping tool, which enables us to run the same measurement at a given position in the coordinate manager. Also, we have an image alignment tool. This enables you to align and overlay images taken on different systems to correlate and look at the inVia results with the results from the other systems. So what I want to do really is, in this example here, we've done some measurements on an inVia contour and on a SEM, and I want to show you how we go through the workflow of correlate and what you can do with this sort of information and why it's really useful to you. In this example, we looked at a mineralogical section and we analysed it using SEM and inVia with Correlate. And the aim of this work was to use these two techniques in tandem to better understand what was going on in this material system. The sample itself consists of a dissolution layer hosting copper sulfide, and this is shown in the image on the right hand side. So we've basically got this copper ore. Then we've got this dark dissolution layer, and then we've got the bulk of the mineral sample. And really we want to understand what the composition of this dark layer is and how does it relate to ore nucleation. And we made some progress towards this, as I'll show you in the next couple of slides. So what's the workflow for this? First of all, we analysed the sample in the SEM. We took images at regions of interest, and we recorded coordinates based on the centre of where those images were taken. We then moved the sample to inVia. We calibrated the SEM coordinate system to match the inVia coordinate system using the coordinate manager. And then we're able to conduct batch mapping measurements in Wire to automate measurements at our chosen positions. Finally, once we've got the inVia data, the Raman data, we can just overlay it directly with the SEM image. So first of all, with inVia, I took a white light montage showing all the positions and the relative references. And here, this is just to give you a real overview of the sample. So at each of these measurement points, a RAM map was taken of around 120,000 points, took around 3.5 hours, and we're doing basically high resolution maps at 532 to try and get the most information we can from the sample. And the nice thing about this this whole setup is, because we're using the batch manager, we can literally set this to run overnight and it will do the measurements one after another, so there's no downtime on the instrument. So now I want to go through the results of the individual positions. So this is position 1, and first of all I'll try and talk through the SEM image. So this is a backscattered SEM image. Broadly, it gives information on the atomic number of the elements in the sample. Where the image is brighter, the element is heavier. So we can see there's differing compositions here. It's not dependent on topography. And we can draw some general conclusions from looking at the Raman data. So with this Raman image superimposed over the top, we can see that this region contains rutile, anatase, apatite, carbon, quartz, and calcite. We can see the darker regions of the image, so regions in this sort of colour, are primarily a mixture of carbon and quartz. The very light regions are apatite, and the middle colour regions, like this region here, is calcite. So we can now immediately make some significant conclusions just by looking at the SEM images without having to do any Raman analysis. We're reasonably confident what we're seeing here. Position 4. We can see quite a similar story. So we've got very good matched light regions being apatite, moderately light regions being calcite. And what we can really see here, and I really want to highlight, is the fact that the backscatter image shows these regions, these darker regions, to be of uniform color. Okay, but the Raman clearly shows we've got some regions of quartz and carbon intermingled, and obviously this has some impact on how the layers are formed and what they contain, and it's really important from a mineralogical point of view. Lastly, I'm going to look at position 3, and here I want to highlight just some of the variation between the two techniques. So again, this region, so we've picked a region here which is primarily carbon and quartz. In the SEM image, we have this black region, and we thought, okay, so what is this? Maybe we can find out from the Raman. Well, what we see from the Raman is not very much, so we see it still matches this quartz, and it probably suggests that it's not different. This could be an artifact from the SEM image, or it could be some surface sort of contamination. Okay. The other thing I want to highlight from this image, which is important when looking at any of these combined techniques, is that we need to be really careful because we're not really comparing apples and apples. So this is more an apples and pears situation. So you can see we've got a very nice match with this region of apatite, but in the Raman image, the apatite seems to extend outwards to the right-hand side, and that's not shown in the SEM image. And that's because the Raman and the SEM measure at very slightly different depths from the sample. So in this case, we've probably got a situation where the SEM is more surface sensitive than the Raman measurement, and it's likely that we'll got an apatite region moving slightly below the plane as we go to the right. So we pick it up on the Raman, but we don't pick it up on the SEM. So combining all those images, just to overview, I'm going to need to point out these aren't to scale, just to give you an idea of what the composition is so we can draw some sort of general conclusions. So what we can see is in the bulk of the sample is primarily made of calcite. You can see that with these blue regions. Okay, if we go to the black layer, we can see it's primarily quartz and carbon, with slightly increased levels of apatite. Although I should point out their apatite is generally present everywhere anyway. But this lets us basically understand what we've got going on here. So the bulk tends to be calcite rich. The dissolution region tends to be quartz rich, and so that's our conclusion. So I'd like to summarize here. Hopefully I've shown you how Rayleigh correlate can be used to combine Raman information with information from other microscopes or imaging techniques, here specifically SEM. I want to highlight here that these measurements don't have to be done in situ. The Raman and SEM can be in different labs, they can even be in different countries. This has the benefit in that you're not fully occupying both parts of the combined system in order to conduct these measurements. So someone can still be working in the Raman and someone can still be working in the SEM to take different measurements. So you're not tying your instrument. This is important if you're doing some measurements where Raman can be quite slow. So you might be mapping a huge area where it takes, I don't know, an overnight run, and in that time the SEM is free to be used by others. From this specific example, I've highlighted that the Raman and the SEM results actually complement each other and provide different information about the sample, and they let us build together a richer characterization of the sample. Specifically, we've looked at the compositional changes in the mineral section. So as we transition from the bulk to the dark layer, there are clear differences. The dark layer, this dissolution layer, is primarily quartz based, and the bulk of the sample is primarily calcite based. There's a lot more work here to basically understand the sample in more detail, but I think we've made a good start, and perhaps the next best thing would be to try a different technique to try and learn a little bit more about the sample again. Now I'd like to go on to my next case study, advanced characterization of rhenium disulfide flakes. Rhenium disulfide is a transition metal dichalcogenide. It's a layered semiconductor, hence a 2D material. It has a band gap of about 1.5 eV. One of the really interesting things about rhenium disulfide is it has a distorted 1T phase, and this is due to a Pearl's distortion. And this means that it has quite interesting crystal properties. It's very anisotropic, and that makes it very interesting for Raman spectroscopy because it has lots of different Raman modes. It's possible to make rhenium disulfide samples using the conventional mechanical exfoliation technique, and you can grow it using CVD methods. In this work, I'm using exfoliated flakes, and these were provided by Lewis Hart and Daniel Wolverston at the University of Bath. And I'd like to thank them. This is a typical spectrum of rhenium disulfide collected with an inVia Raman system. You can see there are a number of different features here. So in this range and going upwards, we have a number of Raman modes. As I mentioned, it's very anisotropic, so you have a large number of modes. However, if we look down here, very close to the laser line, we have these low wave number modes. And these low wave number modes are a result of the individual layers of the rhenium disulfide crystal moving past one another. So you can either have them moving in plane or in shear. And these are responsible for these very low energy modes. And these modes correspond to the number of layers of the material. So as you increasingly add layers to the material, the modes themselves start to become damped, and that changes their energy. So this is an interesting way to identify layer number in 2D materials, and it's something we're going to talk about more in this study a bit later on. So one of the challenges when dealing with 2D materials is the really common way of producing them is mechanical exfoliation. But unfortunately, this produces only very small amounts of material. And this can be challenging because you need to find the right layer thickness, and the exfoliation production method gives you a very large range of thicknesses. So the standard method is to take the flakes, deposit them on a silicon dioxide substrate, and this allows you to visualize the thin layers, so you get some optical contrast. However, it does mean that someone has to spend a lot of time staring down a microscope looking for the layers, and you still can't guarantee that you've produced them, so it can be like looking for a needle in a haystack. Alternate methods for measuring layer number are Raman spectroscopy. This is deemed as the gold standard, so this is very useful because you can do it to a high spatial resolution, and you can do it with high speed and accuracy. So something like graphene, the standard technique is you find a flake, you do Raman on the flake, you determine its thickness, job done. Other techniques are used. In particular, AFM is used to measure the flake thicknesses. AFM is normally used to calibrate the Raman measurements. So basically, doing AFM on very large areas of material is not that practical. So normally what people would do is, for good science, they would find some flakes of varying thicknesses. They would determine the thicknesses using the AFM directly, and then they measure the Raman at those locations to actually calibrate the thicknesses. And there's loads of work in literature with this being done, and it's very easy then to go back and use the Raman, which is the faster method, to determine layer number. In this work, I have done this. So I use two instruments, a Brooker dimension edge AFM, and I use peak force tapping to identify the layer thickness of some rhenium disulfide flakes. I then used a Rayleigh inVia contour Raman microscope equipped with Eclipse low wave number filters. This enabled me to measure the Raman bands even very close to the laser line. The same system was equipped with Rayleigh scattering filters, which I'll talk about later as well. So all these experiments were done on these two systems. So this is a really nice way for me to show off the Correlate tool. So I have two different instruments with two different images, an optical image and a peak force image, and I can use the Correlate tool to combine the two. And here you can see that on this slide. So what I'm able to do is basically transform the two images, so I can align them, I can stretch them, rotate them, translate them, etc. So I can get a really good overlay. And I can then use those images to define Raman maps. I can superimpose the Raman data on top, so I can see where I've measured relative to two images, etc. So it's a really, really useful tool, this Correlate tool. I talked about measuring the thickness of the flakes and working out their layer number. And a really nice way to do that is to use the AFM images. So here I've taken the AFM image and I've just measured the cross section running across this line, and that's shown here. And I can just look at the change in height from that and the substrate. Single rhenium disulfide layers have been measured in literature. They have a reported thickness of 0.66 nanometers. Here I'm measuring approximately 0.9, so a little bit bigger, but not so bad. So now I can use this correlated setup to measure the thickness of the rhenium disulfide at each position just by looking at the AFM image and measuring the topography. And I'm able to do that and overlay it on the image. So I've got regions of 1 layer, 2 layers, 4 layers, 7 layers, 8 layers, and 9 layers on this particular region of the sample. At each of these points, I can tell the Raman system to take a Raman measurement. And I can then look at the spectra to see what's going on. So here I'm focusing on the low wave number region, and you can see for a single layer of material there is nothing in the low wave number region, and that's because there's one layer and there are no other layers of atoms to move against it, so you don't see any bands. There are, however, bands higher up, so I can still confirm there is rhenium disulfide here. And what you can see as we go to higher and higher layers, we have bands. The band position changes, and broadly a pattern is basically they start to move down to closer to the laser line. So you have this like trend coming inwards, cutting in. But what we've got here is we've now got a really nice tool where we have these very distinct band setups for different layer thicknesses, and we can use that to actually image quickly across a whole sample and determine what the layer numbers are in a given location. And that's what I'm going to show you on the next slide. So here I've moved to a different flake, and you can see the flake's quite small. I've just conducted a simple Raman map across the flake, collecting around 250 points in about 10 minutes. And I'm able to very quickly, using DCLS, direct classical least squares, determine how thick each of these regions is. So we can see we've got a 1-layer region, a 4-layer region, 5-layer region, a region where we have between 6 and 10 layers, and a region where we have 10 plus layers. And the reason we have this 6 to 10 layer section is because there's not so much deviation in the bands in that change of thickness. But the important thing here is we're primarily interested in a few layers of material, and we're able to identify whether we've got 1, 2, 3, 4, 5 layers quite easily using this technique. So this is a nice tool. We can image the areas, we can find out how many layers we've got, but can we do any better? And the answer is yes, and I want to talk to you a little bit about the next technique, which in this case is Rayleigh scattering. So throughout this talk, we've talked about Raman scattering, which is a very weak form of scattering. Rayleigh scattering, the elastic part, is much stronger, about 10 million times stronger. So it can very quickly, you can do a Rayleigh scattering image over a large area. The negatives of Rayleigh scattering is it doesn't provide the chemical information like Raman does. But it can let us identify regions of different material and structure. You don't need very much laser power to do Rayleigh scattering because it's so strong, so it's inherently non-contact and non-destructive. On an inVia contour, we're collecting data rates at greater than 1,000 points a second, and we're able to map very large areas to find flakes. So in this case, we can just do the maps very quickly. As a quick aside, this is a Rayleigh image of upper esophagus tissue, and here you can basically see from the Rayleigh image, we're able to get really good contrast on different features in the tissue. So going back to our rhenium disulfide now, we have done a Rayleigh image of the whole region. And this is very quick. We're able to collect 230,000 spectra in approximately 5 minutes. So super fast. We get a contrast image like this. So here, red regions are strong Rayleigh scattering regions, dark regions are weak Rayleigh scattering regions. And what we can see here are a couple of things which are of interest. So the first thing is our substrate is primarily blue. It changes to a slightly purplish as we go through. This is because here we're looking at the intensity, so any deviation in focus is affecting our Rayleigh scattering. So when doing Rayleigh scattering, it's really important you maintain sample focus. The other thing to note is our rhenium disulfide layers. In the regions where we know we've got thin layers, it tends to be darker than the substrate. But as we go to regions with more and more layers, so I think we've got 4 and 5 down here, you can see that it becomes lighter than the substrate. And when we basically go to very thick material, it scatters much more than the substrate. So this is interesting, and this highlights the need for a bit more thought and a bit more study here. And we'll show that in a minute. But what I want to show now is the correlation again. So now I've got a triple correlated image. So I have the optical image overlaid with the AFM image, overlaid with the Rayleigh scattering image. And on top of that image, I've got the points where I've collected Raman data. So again, this shows that you can basically collect all your data in one work package and do the analysis in one chunk. So just to summarize now, just looking at the Rayleigh scatter data and the Raman data. So here I've shown the Rayleigh image. We've overlaid on it how the layer thickness is. So again, we can see in these regions we've got this dark region, we've got 1 layer, 2 layers, still reasonably dark with 4 layers, but once we hit 7, 8, 9 layers, we're much brighter than the substrate, ultimately going to very bright colours for very thick material. And this change in reflectance we can basically understand using a model for optical constants, and I'm going to cover that on the next slide. So what this highlights really is there's a bit more science to understand here. So we modeled these results using a theory of stratified media, as treating the flakes as dielectric film. So very briefly as an overview, so what you do is you take each of the layers in the sample. So in this case we have single layers of rhenium sulfide with a difference number, and we place them on a layer of silicon dioxide, and we plug in the appropriate thicknesses, refractive indexes, and we just look at the contrast between the layers. So that's the change in intensity based on layer number. And what we can see here is that our model reasonably well matches the experimental data. Primarily, we've got good agreement where the turning point is. So you can see that before 4 layers, we're seeing a reduction in contrast, and then above 4 layers, we're seeing this turning point. So we see a higher number, which is what we basically see in the Raman image, more intensity. Now, we do have some discrepancies, particularly with the intensities. They're not bad, not so good at higher layer number, but obviously we don't have a huge number of points anyway. And it's likely that one of the discrepancies here is basically the change in the refractive index of the rhenium disulfide. So we were able to get values for refractive index from literature, but we suspect there's some polarization dependence on this refractive index, and those sort of values were not reported at the time we did this model. So now I'd just like to summarize this case study. So we've used a number of techniques to analyze the rhenium disulfide flakes. We used eclipse filters to measure low wave number Raman bands. We also did Raman mapping using these filters. We did co-localized measurements with a dimension edge AFM, and we used this to get the sample topography and correlate flake thickness with the Raman data. On top of that, we then used the inVia to do Rayleigh imaging on the sample on the same region, and we were able to combine all 3 of these sets of data together to enable us to basically better understand the rhenium disulfide samples and identify quick ways for us to determine layer number. And the variety of techniques used here really highlight the power of inVia as a research tool. There are not many instruments and software packages that would let you do this as its own, and this is this correlate tool is a really powerful tool for doing combined research and for speeding up your scientific analysis. So now I'd like to conclude. In this talk, I've highlighted the benefits of using Rayleigh Raman spectroscopy systems in combination with other techniques to provide additional information, ultimately helping solve complex scientific problems. I've demonstrated the power and the versatility of inVia and really shown you the vast number of techniques that you can now combine the information with in our software. So this new correlate package enables you to combine your Raman data with data from any other imaging technique, so it's really powerful. And finally, I hope that I've shown you some good science today. Using these techniques together has helped me solve problems, and I hope that just some of the concepts I've used may stimulate you to do some more combined work in your future research. So I'd like to thank you all for listening, and I think we're going to open up to questions.
So sorry about those technical issues. I'm just going to get into a few questions that we've had. I think we covered a huge amount there, and that was the final example we had there on rhenium disulfide, a really interesting example. So some of the questions that we've had coming in. So there's a question here about LiveTrack and about looking at topography and what some of the larger materials we've looked at are. And I think that's really interesting because so often we think of Raman, we think of that as a microscopic technique. looking at very small materials at high resolution. Of course, that's a really big part of the work we do, looking at things like carbon nanotubes, nanoscale materials sometimes as well. But as we've got faster imaging, we've got this new topography method with LiveTrack, we're able to really scale up the work we do, potentially looking at lower spatial resolution as well, which definitely has its place. So some of the materials I've looked at, everything from sort of large wafers, where you can get bowing of the wafers and maintaining high spatial resolution is actually really important. But there's other materials as well. So I've actually got one of those materials. I keep it on my desk because it's quite an interesting one. So looking at something like this. So this is an ammonite fossil. So this is collected from the UK. But basically, I've done some Raman imaging on this ammonite fossil, and that's a mollusk. And what's interesting about this is there's a lot of surface topography there from the ammonite itself. Those ridges are called septa, and they actually are a really good example of how you can use correlation of topography with the Raman information, where there's a change in crystallinity there, where the septa are in place. And that's exactly the sort of information we want, with our different techniques we use, and really getting that nice complementary information to help our science, help our understanding of the materials we're looking at. And it's a slightly different application there, but I think it kind of complements what we've heard here really well in terms of the sorts of things we look at. Tim, any other materials in terms of using LiveTrack that you think are kind of relevant to that question?
Well, first of all, I'd like to apologise to everyone because it seems like I was disconnected before the talk finished. I managed to go through it myself before and finish it up, but I guess it didn't broadcast, so I want to apologise again. I believe I got to the rhenium disulfide example, and I think from what I've been told, I was one slide away from finishing where I lost audio, so you did get most of the talk at least. Okay, going back to the LiveTrack example. So as I mentioned in the talk, I really like this Raman board. It's a really crazy thing. I don't know why you do Raman on it, but when my colleagues were sent this sample and they measured it, and it looks really beautiful in the image. We've done other Raman measurements on interesting samples. So we've measured things like coins. I've measured an aircraft turbine blade. So you can imagine now you've got a really large object. And again, basically, as long as you can fit it under the microscope, you can track the whole gradient in that object. So it's a fantastic tool for that. More realistically though, what people tend to measure with LiveTrack are things like wafers, or one of my interests is 2D materials. So I measure a lot of graphene on copper foils. And with these types of samples, because the copper foil sort of undulates, it makes it very difficult to keep in focus. But with the LiveTrack, it just absolutely follows the surface the entire time. So you get really nice images with high mag lenses. So it's really quick.
Right,
Carrie,
and then another question. Yep, another question was in regards to the battery. What is the dimension of the sample?
So I think this refers to the battery example. So the collection area for the Raman was 125 by 125 microns. The sample itself, I'm struggling to remember. I think it was about 10 millimeters square. So it's basically a square electrode. I can't talk for its provenance, but it's basically a thin film type electrode.
Okay, and then is Raman instrumentation easily portable from SEM to AFM?
Okay, so this is a bit more of a trickier question. So with the AFM integrations, we tend to have a direct coupled system. So you'd sit the Raman system on the same anti-vibration table as the AFM, and that gives you the best quality data, so the best throughput, and also ensures that you stay on the same spot. Now, for our SEM integrations, we use a fiber coupled approach, and that's because you can't really set a Raman system next to a SEM and maintain all the balance and the coupling. So from that point of view, you could have a system which is connected to both an AFM on the same bench and a SEM several rooms away, or even in the same room. And so you've got the choice when you set up the system. If you're interested in more portable Raman systems, I know 3 weeks ago, so the previous webinar we did was on our new Versa Raman analyzer, and that's a portable, transportable type analyzer. And I think Tim Prusnick gave that talk, and he gave quite a lot of details about the different techniques we can couple to with that system. So if you're looking for something truly portable, that's a better system than this one we're looking at today, which was the inVia primarily.
Yeah, there's definitely some logistical issues, in terms of just the space, and whether it's an SEM and an AFM, even getting those in the same room, can be challenging at times. And I think, the Correlate module is a really nice solution to that as well, where it can be, like Tim said in his talk, you can be in different countries and still get that complementary information. So, I think if you can have it hard coupled, or if you want to use them independently, then you can do that as well with our software.
Okay, Carrie, next question.
Sorry about that. Is TERS usable as a regular analytical tool?
Well, TERS is a very challenging technique. I think it's come on leaps and bounds over the last 15 years. And we've been selling TERS systems for, well, since some of the first commercial systems we sold were TERS systems. But the situation is that it's probably not ready for continual analysis. There are certain sample types which work quite well with TERS, but the repeatability is still a little bit in question. So when you work on these very small length scales, so you're down to the nanometer length scales, I think the science becomes a lot more interesting. And what you begin to realize is the variation on these length scales is very large. And it's something that when we're kind of doing routine analysis, we're more sort of looking for an overall average answer, and TERS can throw out really sort of like very infinitesimal results, which make for interesting science and interesting understanding, but not so good for just getting simple analytical results.
Perfect. And then the next question I think is more for Tim Smith. To upgrade to wire 5.3, can you use Correlate with regular mapping stage and no autofocus?
Yeah, absolutely. So the autofocus is part of our contour inVia Raman system. That gives you the ability to track the complex topographies that Tim talked about. But yeah, you can use Correlate with standard sort of conventional mapping, and it works just as well. You can manage also the Z positions within Correlate as well. So if you want to independently look at different Z positions for the different particles, you can do that as well, and that's something you don't need LiveTrack to do. LiveTrack really comes into its own with the mapping. If you want to do the mapping over complex systems, or you've got dynamic systems in place as well. But yeah, so you can definitely do that.
Okay, so one thing just to add to that is that Correlate is a free upgrade to anyone running wire 5 or later. So if you have any version of wire past wire 5, you can upgrade and get Correlate for free.
Perfect. And then since Tim Smith, you mentioned live tracking, the next question is they are curious about the comparison between the speed of the new live tracking feature compared to the older autofocus.
Right. So historically, we've had an option called LiveTrack, called PhotosTrack, and PhotosTrack works before you do any data collection. So we're basically stepping through, we're looking at the change in the focus of the laser. Once that's been optimized, then we can go off and collect our Raman data. LiveTrack's completely different. It's effectively a live method where we're continually giving this kind of live feedback, making sure the sample's always in focus. That means we don't have to interrupt our Raman data collection, and that really means that particularly for the mapping type measurements, you don't have any overhead, and so you can go ahead, do your mapping, and you're not spending any extra time. PhotosTrack, there was quite significant time delays between each data collection, so a real difference there.
Okay, and then keeping with the live, sorry, go ahead.
Sorry, Carrie. So really what I'd say there is if you're looking at sort of the old PhotosTrack technology, compare it to LiveTrack, with LiveTrack, you might see a factor of 100 to 1,000 times speed increase. It's phenomenally faster, and you can basically use LiveTrack for fast mapping, sort of 50 milliseconds a point, no problem at all. And with the PhotosTrack method, you might be up to 10 seconds a point to give you a comparison, so you can kind of see the differences.
Perfect. All right, next question is, can the contour system couple to SEM and AFM, or can the inVia reflex can also couple to them?
Okay, so I'll handle that one. So yes, if you wanted to, you could couple an inVia, either a reflex or a contour, to both an AFM and SEM. So they use separate paths. As mentioned before, normally the AFM is on the same optical table as the Raman system, and the SEM, because it's fiber coupled, can be up to 100 meters away.
Okay, what is the accuracy resolution of the SEM and Raman map correlation?
Yeah, so I've done a lot of work in Correlate. I was involved in the production of the module itself, and one of the key things is obviously making sure the resolution can handle both very high resolution and low resolution as well. Because of course with Correlate, you might not only want to correlate to Raman, but you can correlate something like infrared, which actually has a lower resolution. So yeah, we can combine all those together. You can zoom in, and certainly to the micron, submicron level, you can get correlation there. In terms of actually moving your sample from one technique to the other, the real key there is to have fiducial markers that you can get to with very high precision. So you want to be able to get to a certain micron point on that fiducial marker, and of course you want those well separated relative to the positions you want to analyze, and that will minimize the error you have in terms of then translating it onto the Raman system. So yeah, certainly able to get sort of micron level precision with that type of correlation work.
Okay, now the next question, I'm not sure when it came in, so it sounds like they were talking about a specific technique. Asking if it's also suitable for imaging, or I'm sorry, yes, also suitable for imaging of tablet and quantify each components present there.
I mean, in general terms, you can definitely quantify material in tablets. You can do that directly from an image, so you can do things like the area as a percentage within an image, or you can get something a bit more concentration related. And yeah, we use LiveTrack certainly. So some of the common uses of LiveTrack are certainly looking at things like tablets, whether they're split tablets or whether they're whole and they're kind of curved and indented. In terms of SEM and AFM, I'm not sure how that links in. So maybe that was more about LiveTrack, that question.
Okay, and then do you think the Raman technique can be combined with a rheometer and then also be used as in situ Raman? Not sure what that means. And it says under shearing conditions rather than, not even sure.
Could you ask that again, please, Carrie?
Yeah. Do you think that the Raman technique can be combined with a rheometer?
A rheometer, yeah. Okay.
Yes, thank you.
Yeah, so
sorry, my Raman technology or verbs aren't very good.
Okay. Yeah, so you can combine Raman with viscous measurements with a rheometer. With that, you don't tend to get spatial information, but there's no problem doing it combined. So we have fiber probe-based systems which you can basically use to point into the fluid as you're stirring it, and then basically determine what the composition is and how it's changing elastically.
All right, I'm going to ask one more since we're almost out of time. What is the benefit to having Raman plus SEM in comparison to correlate techniques that you described?
So I think with the Raman inside the SEM, you can do real-time backscattered electron and Raman data collection. So there's no time delay between them. There's certainly limitations of doing it inside there. Of course, you've got convenience as well. You've got the same sample in the same SEM, so you don't have to sort of take the sample out. But on the flip side of that, there are definitely benefits of using them independently as well. In terms of the flexibility and the versatility of how you go about using the different tools of the Raman equipment. So, for example, inside the SEM, you're really limited on the different objectives. So basically, we're limited to one objective. You don't have LiveTrack. You don't have a lot of the fast mapping options. So you're much more limited there about how you can go about and do your work. So I would really say for if you want to do work inside the SEM with Raman, then that's going to be relatively basic work most of the time. Even with mapping, that's going to be relatively basic type mapping functions, and a lot more of the advanced research information you're going to get by doing them separately. Anything to add to that, Tim?
I suppose the only thing I'd say is if you're doing Raman in the SEM at the same time, you can see if there are any changes to your sample, which can be important in some cases. So you can see, for instance, if you're damaging
Right.
a sample, use it during the SEM analysis. For instance, we see a carbon buildup or something like that. Doing them standalone tends to be better in most cases because it's quicker. You can get better resolution, both primarily spatial resolution, that is, from the Raman side, and having the correlate tool where you can basically bring them back to within a micron of each other means that there's no need to do these, spend a long time doing these high resolution maps in the SEM itself.
Okay, thank you so much, guys. And if you asked some questions and we didn't have a chance to answer them, we'll touch base with you individually to answer those questions. And then just to close, I'd like to thank you again for joining us today. Sorry for the technical issues. We appreciate you sticking in with us here. And we hope to see you again at our next upcoming webinar, hopefully with no technical difficulties. So keep a lookout for an email with information on our next webinar, where Tim Smith will be discussing Renishaw's new particle analyst software. And if you'd like to learn more or have any additional questions or concerns, feel free to email us at usa@renishaw.com, and please follow us on social media, where you can stay up to date on our latest news. Thanks so much and have a great day.